Hi, everybody. So we've been giving a bit of a background about the AML organization, which is the part I lead at GSK. So we're a team that works on very early stage drug discovery and development. So really the targets, which is sort of, what do we make our medicine against, to all the way through to the clinic. Who to give this medicine to, when and why, predicting how to respond, and making sure it's safe and reliable. And we use machine learning for all these different tasks. And as Julie mentioned, we've been playing games with Cerebras since about 2019. So this is a little picture here of a CS2 that was installed in our Philly data center at that time. And we were drawn to this technology because we have an exponential amount of data happening in medicine, right? It's due to the fact of single-cell sequencing. It's due to the fact that we have these large digital medical records. It's imaging, so it's not only clinical imaging, it's pathology. And all these data sets are coming together. But we're particularly interested at that time in building these models on DNA and RNA sequence. And the challenge is they're very large. We just started building the group. And we didn't want to play games of both model parallel and data parallel. And the thing that really drew us to Cerebras is the fact that we can build models, right, that we weren't limited at the time, and back in 2019, of the size of memory on the chip, right? So you can stream both the data and the compute graph at the same time. And so our criteria was, someone must be building this hardware. It has to exist, and it has to exist now. And Cerebras fit the bill. So this is the first machine getting installed. And when we got to know each other, we realized that we had a lot in common because we both had cool conference room names. So they have the war games there memory for us. So I'll start off where we started, which is the sequence learning thing I talked about. So we like to build these models on top of DNA and RNA. And this is the first of one of these models we built. These models are trained on the linear sequence of DNA and RNA, and they have various sort of properties. And what we do is we show the model many examples of these sequences with their various contexts. That means we can ask various questions, like, is this gene expressed in this cell type? What genes are turned on and off in this cell type, right? Which isoforms are built, et cetera. And that's really important because we use these models now to design drugs that are based on mRNA. So a lot of you have heard about mRNA in vaccines and vaccine technologies. Well, mRNA, or oligonucleotides as we call them, are very useful for a new class of medicine. So in your mRNA, which we all know, DNA makes your mRNA and goes to protein, there are various sort of splicing forms, right? So there are isoforms, and there chunks of that message may be dropped out and spliced in to make a slightly different protein, right? And so there are cases of disease where one isoform is toxic, the other one isn't, right? Or we want to modulate things. A lot of our medicines we make usually block information flow. Small molecules may stop a kinase working, or an antibody may stop something binding. With oligonucleotides, we can actually deliver them to the cell and increase and make a very specific type of protein. Make more of splice form A over B, which can affect the effect. So all these models that we built in Cerebras, they get used to design these, right? They get used to say, hey, which splice form is going to be dominant? If I design my oligo here, will it stabilize the splice form I want? Can I destabilize the splice form I don't want? And these sorts of things. So we use these right now in production to design oligonucleotides. We have a huge active learning program. We recently acquired a company called LC Biotechnologies, purely for the sake of generating data that feeds back into these models, and these models are being trained in Cerebras, right? And we've moved on from just training on one single CS2 to multiple ones. So on that thing of moving on, right, I'll talk a little bit about an LLM we started building. So our LLM is called Brad, and we started building an LLM for two reasons. Like many companies, we had a giant knowledge graph, and we had a whole sort of classical sort of NLP thing for reading papers, extracting out the facts, and working out the associations and things like that. And around the sort of GPT 3.5 time, I was playing with that and realized that I could use the LLMs to parse out and extract the facts. And it's something I just hacked together on a Saturday morning. And it was better than our 2-year built, really tuned kind of NLP pipeline. But at the time, we realized a lot of these large language models, they have some knowledge of science. They're not bad. But for what we do, the details matter, right? And this is just an example. This is GPT-4. Like, well, what do you know about P10? It knows basically like half the problem, right? It doesn't know everything that's going on in there. And if we want to start using these models as tools, right? We don't use them as a database, but if you want to ask them to reason or summarize and things like that, they need to have a lot more information in them. So we started then basically building a series of these models. So we started off in 2013 with a smaller model, which is our sort of our first era sort of these model. And we're basing these on the Llama stack, right? So we're taking the Llama series of models and having like the full open weights is very, very important for us as a company, right? So we know exactly what's going there. We can fine tune it. We can push it various sorts of things. And you can imagine that we have a lot of scientific data, right? We have licensed a lot of data from different sources. We have a lot of internal data as well. GSK is a 300-year-old company. We've been doing science for a long time. We have lots of clinical trial data. All this stuff comes together. So this is all going into our models. And so we built a series of these models sort of increasing in size over time. And our kind of process is we take the models, we do our pre-training, we do fine-tuning on a lot of different tasks we care about, not just the canonical benchmarks. We also make an instruction version of these models, and then we kind of deploy them in production, and they go into our agents and our tools, which I'll talk about. And so the first set we started off, I guess the latest model, is running on 16 CS3s. It only took 27 days to train, and I know that seems like a long time. Compared to how long it used to take these models to build, that is a very short time. And we basically found that, okay, we can beat the Llama 3.1 base models and the instruct models on some key benchmarks. And it's certainly better on the benchmarks that we've constructed our own internal benchmarks, as you might imagine, that we really care about. And so we're getting about sort of a 7% performance boost with a bit of training for us. And that doesn't seem like, well, that's a lot of effort for 7%. But for the things we care about, that 7% really matters. So I'm just clicking forward here. Look, it wouldn't be a Cerebras talk if I didn't show you some loss curves. There's a lot of people involved in this. What we actually do in this case is that you can imagine the scientific literature is coming out in phases. What we're actually doing is showing these models in our pre-training of time slices of the scientific literature, because we want to build these models up and saying, well, here's the first, here's the earlier sort of work all the way up to 2007. Here's the next set of data. Here's the next set of data, and then we're using the new things to predict. Can we now infer discoveries that would have been made later on from those different checkpoints or things that will happen in the future. So we're slowly showing this model more and more data over time. And what we find is that we can build a model which is pretty good and these are the high-level benchmarks that we care about, right? So we haven't obviously improved on the general knowledge and the general aspects of things, but you wouldn't expect that to happen. But we have improved on some of the science questions we care about. And more importantly, we're getting a lot more improvements on Lab Bench, right? Those of you who may not know, Lab Bench is a new benchmark that came out from the friends, the good people there at Future House. And we're starting to use a lot of these LLMs as agentic workflows in this sort of AI science and other types of things. So we're now trying to push these things into different directions. So we're pushing very aggressively on fine-tuning and building models that will work on these sort of benchmarks and others. We find the benchmarks are a great way of understanding that you're going in the right direction for training, you've got something good. But what really happens, you still have to create your own benchmarks. So we've created a whole set of benchmarks at GSK, and we will probably open source some of them. A lot of them, unfortunately, use our own internal data, so it's very difficult to open source a benchmark. But we find that, in general, on average, Brad v2 is beating all the other models that we've tested. We haven't tried, obviously, with the Llama 4 series because they weren't out at the time of work we're doing this, but we'll be quite excited to look at that. But things we care about, like answering questions about protocols, designing sequences, work at cloning, the model does well at those sorts of things. We found we could get some further gains by working a bit with the Cerebras planning optimization, which is a nice paper that came out. And so we're using that to do sort of, basically this is effectively doing, reasoning type actions on that. You can eke out some small gains with that as well. And we're still kind of pushing on this right now. But where things become really interesting is when we start to use some of these models in agentic workflows. So obviously you can imagine there's a lot of science happening in the world, right? And we launched the sort of the RAG summarize my paper thing when these sort of things first came out, using OpenAI and Azure. And that's great. I actually, that was the first use case. Here's a complex paper. Let me ask a few questions about it. Or in my case, here's a boring thing someone asked me to read from other boring parts of my job and let me know what I need to know about it. But really, often when I'm trying to think about a topic, there's lots of papers to read, right? And helping you speed up or summarize one thing means you still have to go through this thing where you're exploring lots of papers and you're trying to synthesize it into one big consistent piece. And you might come up with a hypothesis or some idea you're going to go and talk to someone about. You get some more knowledge. You might get some experimental feedback. We might get a clinical trial readout. It all goes back into the pot, and we have to think about things again. So just summarizing documents still pushes the hard work back onto the human. So what we really want to do is summarize the literature and present a compute view back to our scientists, right? So going on beyond RAG, so let's have something that reads all the papers for me. So we have hundreds of millions of documents in a store. And I can ask a question like, well, what are the key molecular mechanisms that underlie the progression of COPD? COPD is a chronic obstructive pulmonary disease. And so what we have here is you can see it's searching for memories. These systems also have memories for chats and things like that that you can put in it. In this case, we're just looking at the literature data. And it's going to go create the query, retrieve the literature, summarize it, and give us a response. This is happening, and this is a real-time video. This takes about 30 seconds to run. And look at this. Interestingly, LLM is no longer the bottleneck. The vector store is. Right? This has been 20 seconds on that vector store. The rest of it on the LLM aspect. And just to show it's a bug, we can say, well, now I've got my next question. Well, what are the strategies for why this anti-protease balance might happen? It'll then go away, pull in the literature, query it, summarize it. And it's really important for us that we have the grounding. So I can go back to the original papers, and I have the source there. What you might see there, you have this reasoning, and we have this certified response. Right? So maybe we can dig into that a little bit more. Behind the scenes here, we have a whole kind of actor-critic type process that's running. So you can see I'm clicking on this is a slightly different dev version of this deep research. I'm putting in my question. I like to use the literature corpus only. And then you see it actually, in this case, it's running running with Cerebras again. We're doing planning with a more advanced model. Okay. We're constructing our queries, and we run this loop where we get information back in, it comes back in, so we fork off a bunch of different worker agents that work sections of documents, they bring their summaries back in, right? And then we have a query that looks at that and says, actually, did you do a good job or not? And also that feeds back into our planning agent, back to R1, who then updates the plan. And we run this loop as many times as we need to our final judge argument decides that, yes, this is a reasonable response to the user's question. And so obviously we fine-tune that judge, right? And that's actually what happens all behind the scenes before you see that answer returned back to the scientist, okay? And this is great because now not only can we put this over the literature which is emerging, new papers that are coming in, the archive and things like that, but also the internal corpus. And you can see that what we discovered is when we do model serving at speed, right, it matters. Previously, these things, you could go off and do this sort of stuff, and it would go off, and it would take a few minutes or longer than that, and it'd come back. And people would use the system one or two things. They might use a system, just for some occasions, they would go and do things, because no one wants to sit at their desk for like 5 minutes. When we switched to fast inference, right, the engagement, the use cases, people started using this more and more, right, and for all these different systems. So the speed really matters for that user experience, right? And I think many of you, have forked off deep research queries and, you can close your web browser, I'll keep working, right? Well, we actually don't have this anymore. Like, you don't need to close your web browser, it's right there. And that iteration cycle, I care a lot about speeding up the iteration cycles of GSK, so how fast people can think, right? And get to the next idea or assimilate new information. That's really key for us. So we can take this one step further. And I'm going to talk here today for the first time really about our AI scientist program. So like many people, I mean, AI scientists are an interesting thing that are sort of appearing in a bunch of different cases. Ours is called Cogito Forge. We really have sort of two ways we can think about it. Either a human may have a particular hypothesis about something which they want to test, or you can give a system a goal, find me new things for COPD treatment, right? We then have to think about ways of representing that hypothesis. And this is a challenging problem, right? You can't do it in first order formal logic, but you can't just be free-form natural logic either. You need to represent it in a way so you can work out, well, if this is true, what would that mean, right? What does that, if this is true, what does that entail? Once I know what it entails, I can then start to generate evidence. We generate evidence for and against our particular hypothesis, and we do this by like using what we call data in the loop. So we have these very, very large data sources, just like the Mayo data source that you just heard about, or internal clinical data, or literature data, or experimental data sources that we can go and query, right, with tools to produce evidence for that. You also could take it one step further, and we do this in a very narrow domain, which is, okay, if this is true, write me a protocol, talk to my lab robot, and have the lab robot go and generate the experiment and pull that data back into me, right? Data in the loop is obviously a lot faster than lab in the loop, because biology takes time, cells take time to grow, and data in the loop is a lot cheaper than lab in the loop sort of stuff. So we tend to do data in the loop first, but you can have a system that goes through, generates this data, ranks the hypothesis, and it has sort of a stack of working hypotheses with the evidence, right? And this can feed it back to the human, who can then think about the new idea, or we can run this autonomously, whereas new data comes in from the world, and that's data from the scientific literature or experiments and things like that. We can say, well, does this allow us to produce any new data and change the ranking and the information for the system? So if we put this together, and this is an example of this running using some graph databases under the hood to help kind of think about the thing rapidly, and then actually we'll generate a hypothesis. So this is our biological association graph and verification framework. Okay, so it's going to load in a bunch of background data so it can quickly formulate hypotheses. You can see we're going to use Cerebras as our provider in this. And what we have here is a series of criterion scores we think about as we come up with biological hypotheses, right? Is a consistent pathway? Can we make something against this? So I might put in my query, and this is like, I'm literally saying, I've seen this gene in COPD patients. It's probably got something to do with it. I'm not, go and figure out a hypothesis to me and think about this, right? Rather than me put in a manual hypothesis. So it's going to generate some structured hypothesis from the context, right? And what's interesting, the context is behind the scenes. This has gone and pulled in all the information we know about COPD, SERP1A, from these genetic interactions of things, and given this to R1 to start reasoning and create some hypotheses, okay? And it's come up with a pretty good hypothesis, okay? It must be causally related in pathogenesis of COPD. Its role is antiprotease, okay? So here's a disease condition, psoriasis at the bottom here. And it says there's this type of hypothesis. We have different types. There's a causal hypothesis. And then it's thinking, well, these are my key mechanisms, right? How this is working. This is pulling in from the literature and other sources. Now I'm going to make my testable predictions, okay? I could do these various types of things. An analog hypothesis is an analogous hypothesis where someone else has put something similar in the system, so I can know if is anyone else in GSK thinking about this. Okay, I have my uncertainties, and then what I'm going to do is, like, here's my final statement. I'm now going to go off and actually call tools and test this. And you can see some of the data we've used behind this that we have that we've used to construct this hypothesis. So what things are interacting with this in the literature, what's interesting is from genes as well. And you can see there's a lot more noisy information in the literature that you could possibly test, right? So now we're going to go and verify this. So now it's pulling in those documents from literature. It's pulling things from other sources. It's going to summarize them all. And we use, again, we like to use these frameworks where we have critics, right, for and against, okay? This is calling other tools and put it all together. Okay, so now you can see evidence for the hypothesis, okay? And interestingly, evidence against the hypothesis and an overall assessment. If you brought this to me, I would say, as a hypothesis and something to do with programming, it's kind of mid, right? Yes, there's something there, but it's not really strong on all the sort of scores, right? Is it novelty? No, it's well known, right? Could we expand it? Is it really going to change standard of care? We care about these sorts of things, And you can actually get all the evidence behind this from this critic of how it led to these scores and these conclusions and things like that, right? So this is a fully working, right, agentic AI as a system. Okay, you can see this is a real demo, but because I haven't got my titles here for all my documents. And we combine this in a Bayesian process, right, to give you this sort of final score, right? It's looking at a whole bunch of documents. Those summarizations are all the summary documents from the evidence that's put together, right? And it's happening really quickly. And we're running all this inference on Cerebras as well. And again, that speed matters, because now people can explore these types of things, and we can put them in the loop, right? And when we have lots of hypotheses and lots of things running over many disease programs across, GSK, right? We work in a lot of different areas, from infectious diseases, vaccines, respiratory, oncology. these tools have to run fast, right? And they have to be able to update themselves as new information flows into the world. So I will leave it there. I'm just going to thank some of our dear friends at Cerebras who actually did this work. Thanks all.